Sampling should occur at the time and place of highest risk. For instance, sampling immediately after intense cleaning and sanitation would most likely result in a negative test, with very little insight into the risks for contamination. However, if you wait a few days after cleaning and then perform the sample, you will get a more accurate picture of risk for that area. Indicator organisms are microorganisms whose presence indicates the probable presence of pathogens. Pathogens are disease-causing microorganisms, but they are harder to detect sometimes than indicator organisms because they are smaller in number, usually. Because indicator organisms grow under the same conditions as their pathogenic counterparts, we can assume that if the indicator organism is thriving, so would its pathogenic counterpart. Also, indicator organisms are often less costly to test for than pathogens, due to their not being pathogenic and therefore not requiring containment facilities or more expensive growth media. There are many tools that can be used to collect samples. These tools will differ from facility to facility and within the facility zones. These tools must be handled and used per specific instructions and properly labeled and shipped to a lab for analysis. Some examples of these tools could include swabs, sponges, air sampling, food residue, dust scrapings, Rodak plates, agar plates, rinses or water liquid samples, or ATP bioluminescence assay kits. No matter the tool, you should always wear sterile gloves when collecting samples to prevent cross-contamination. The sampling team should be well trained in sampling procedures and understand the importance of collecting an accurate specimen. Once samples are collected, they are then labeled and shipped as per the sampling procedure. Labels usually contain the date, location, device, size of sample, list of the testing requests, date submitted to the lab, and any other identifying information to show where the sample came from and how it was collected.